on her shoulder, then lifted her chin and marched to him. No way would she allow him to know she was anything less than perfectly comfortable. When she sank onto his bench and pulled 2 bottles of water from her bag, his eyebrows spiked. It's a warm.
Okay, go get her. No, I just took her out. It's probably been about, I don't know, 20, 15 minutes. First, any questions or concerns? Can you hear me?
Yeah, I can hear you.
Good. All right.
I actually do have a question. I was just wondering when we can actually see the answers for exam 1, like the ones that we got wrong.
You know what? Let me see if I can set it up right now.
Thank you.
Here. Mom, would you bring her back to me if she's just going to jump in there? Guys, give me one moment. I'm going to go grab my dog, my teaching assistant.
In progress.
You know what? You should have been able to see it by now.
I can double check, but I thought I did the other day and I still wasn't able to.
Can you share your screen with me after class and show me what you see? Rachel, can you do that?
Yeah, I can do that.
Okay.
I have a question.
Go ahead, Lakeisha.
I just wanted to ask, did you send our midterm grades to us?
Yes, I don't know how you access them, but they've been out for a couple weeks.
Oh.
I don't, that's a question you probably need to ask the Hack IT Tech if you can't access them on your grades.
I don't even know where to start to look though. I didn't know where to go.
I don't know either.
Okay.
I only know how to do things a lot of times from my end. I don't, because I've never been a student at Hack, I don't know how to access your grades from your end. But that's a question you might want to ask Hack IT Tech.
All you have to do is go to My Hack and then click, hold on, which one is it? Registration and records at the top. And then you'll be able to see it all the way on the right side of your screen.
Oh,
Thank you.
Welcome.
sorry, just a sec. So we will finish chapter 12 today, and we will get a pretty good jump on chapter on the spinal cord. You'll see it's not as long of a section.
I'm sorry, Professor, which chapter is this?
This is chapter 12.
Thank you.
So we will finish chapter 12 today, and we will get a very good start on chapter 13. We might finish it. I don't know, but you'll see that it's not as long. So just reviewing just a little bit here. Last time talking about the nervous system, we talked about action potentials, local potentials, and myelination versus the non-myelinated axons. So with a given neuron, there's an input zone where there's many signals being received from other neurons. These are received generally through the dendrites and the cell bodies, sometimes through the axon, but generally through the dendrites and the cell bodies. Then there's the trigger zone, which is also close to the axon hillock, usually about the same place, axon hillock trigger zone, that whether or not an action potential will initiate it or not, that will go down the axon. The conducting zone is going to conduct action potentials in an undiminishing fashion over long distances. It can be over a meter or long, 1 meter or longer. At the end, the axon terminal, or the terminal arborization, is going to release neurotransmitter that will then ultimately influence other cells. So now we're going to focus on the synapse. What's the synapse? The synapse, I like to think of it as like a snap on a shirt. The synapse is where one neuron joins with another neuron or another cell. It doesn't always have to be another neuron that the first neuron is joining with. It can be another cell, because a lot of times it's just an effector cell. It doesn't, like a gland or a muscle cell. So signals arrive at the presynaptic neuron via the axon. And we think presynaptic because this is a neuron that's before the synapse, which then will release a neurotransmitter onto the postsynaptic neuron. So the presynaptic neuron before the synapse, the postsynaptic neuron after the synapse. In between these two, there is what we call a synaptic cleft. But the presynaptic neurons can synapse with a dendrite, the cell body, or the soma. Remember, that's another term for the cell body, or the axon of a postsynaptic neuron. One given postsynaptic neuron can be involved in the synapses of at least 10,000 axon terminals. Or, well, let's put it this way. One postsynaptic neuron can be synapsing with up to 10,000 axon terminals with other presynaptic neurons. So let me reword this one other way so you understand the gravity of this. One postsynaptic neuron can be synapsing with up to 10,000 presynaptic neurons. If you think about that, this is how neurons integrate information. This is how they receive information. This is how they process the information and make a decision how to respond. So in the cerebellum, which we'll talk about in chapter 14, in the cerebellum, one neuron can have as many as 100,000 synapses. So I said up to 10,000 axon terminals can synapse with one neuron. In the cerebellum, one can synapse up to 100,000 with different incoming neurons. We have two different types of synapses. We have electrical synapses and we have chemical synapses. Electrical synapses are where adjacent cells are connected, joined through gap junctions. What are gap junctions? This is something we covered in chapter 3. What do you remember what a gap junction is? Somebody look back or Google it for me. What's a gap junction?
Direct cell-to-cell transfer of ions and small molecules.
How do they do it, Rachel?
Intracellular channels.
Yep. So these are little tunnels that connect individual cells. They're what we call them is they look like, bear with me, I'm going to stop sharing for a minute and put up with my 3rd grade writing or kindergarten writing on this whiteboard. But here's one cell membrane, here's another cell membrane. Oh no, that's not what I wanted. Okay. One cell. Oh, this isn't working very well. Here, just one cell has little channels in it. So one cell adheres to another cell via half of the tunnel. Another cell, and these are actually almost in direct connection with one another. One cell adheres. So each membrane comes with half of the tunnel, and then they come together and combine. The halves of the tunnels are called connexins. So they each come together, and this produces a tunnel which allows ions to flow between the cells. So this allows for synchronous activity and quick transmission. These are really important in smooth muscle and in the heart. The most apparent that is easily understood is in the heart, because it's what allows all the heart muscle to contract and relax as one unit. Each of the heart muscle fibers are connected via a gap junction. So they all know when they're supposed to contract and relax, and they do it synchronously. Chemical synapses are where neurons are going to communicate via these neurotransmitters. So electrical synapses, the action potential is going to spread from one neuron to the next via gap junctions. No neurotransmitter is needed. It's extremely fast and it allows for two-way conduction. It provides the coordinated contractions of all my muscle fibers all at once. Can someone mute themselves? The structure of a chemical synapse involves an axon terminal, a synaptic cleft, and a postsynaptic neuron. So this is where the one presynaptic neuron is going to synapse with a postsynaptic neuron, and in between them is a synaptic cleft. The axon terminal contains synaptic vesicles with neurotransmitter within them. The synaptic cleft is about 20 to 40 nanometers. So that's 10 to the minus 9th of a gap between the presynaptic and postsynaptic neurons. The postsynaptic neuron has receptors, neurotransmitter receptors that are going to bind to the neurotransmitter. Questions? In all synapses, because of the synaptic cleft, so an action potential is reached here, the vesicles have to bind to the presynaptic membrane and the neurotransmitter gets secreted into the cleft. The neurotransmitter diffuses over the space and binds to a receptor. And then the activities will be activated within the postsynaptic cell. For this reason, because these processes have to occur before something occurs in the postsynaptic cell, there's always a synaptic delay. So there's a time from the arrival of the signal at the axon terminal to the beginning of an action potential at the postsynaptic cell. Neurotransmitters are molecules that are synthesized by a neuron and released when a nerve signal or an action potential reaches an axon terminal. The neurotransmitters all have specific effects on the target cell's physiology. Most neurotransmitters are released via exocytosis. What that means is that some neurotransmitters are not working via exocytosis. So keep that in the back of your mind for a few minutes here. But most neurotransmitters are released via exocytosis from the presynaptic neuron. There are at least 100 neurotransmitters that have been identified. And most of them fall into 4 major categories. The most common one, and you will see this over and over again, is acetylcholine. Some other ones are amino acid neurotransmitters. If you remember any of the amino acids from chapter 2, some of those amino acids are glycine, glutamate, and aspartate, which means that these same amino acids work as neurotransmitters. Aminobutyric acid is a derivative of an amino acid. Monoamines are amino acid derived neurotransmitters that have had the carboxylic acid removed. These include catecholamines, which includes epinephrine and norepinephrine, they're the major players in the sympathetic nervous system. Dopamine, serotonin, and histamine. Histamine, if you struggle with allergies, I struggle with allergies on a regular basis, and fall is usually a difficult time for me for that reason. This is your culprit, is histamine. One other category of neurotransmitters are purines. Adenosine, where have you seen adenosine before? That's one of the nitrogenous bases of DNA and RNA. Also, adenosine triphosphate, our general ATP, our general energy currency for our cells, also can act as a neurotransmitter. What this means is that different things can act as neurotransmitters or hormones based on whether or not they bind to a receptor and signal a particular activity. It's not the neurotransmitter or the hormone per se that's going to carry out an activity. It's going to be whether or not it binds to a receptor. And what that receptor then activates when that molecule binds to it. So the receptor is what's going to dictate, the receptor is what's going to determine the function of a neurotransmitter. Any questions? Some examples, some other categories of neurotransmitters: gases, nitric oxide, and carbon monoxide. Nitric oxide is what makes Viagra work. Carbon monoxide, you're most familiar with it as because it's a poison. It's what irreversibly binds to hemoglobin and carbon monoxide poisoning. However, it's also in small amounts, it's a neurotransmitter. So gases, nitric oxide and carbon monoxide are synthesized as needed. They're not stored in synaptic vesicles. These are the ones that are not going to be released via exocytosis, because they just diffuse out of the axon terminal. Nitric oxide is also a neuromodulator. Neuropeptides, cholecystokinin, endorphins, enkephalins, and substance P are what's a peptide? Remember what a peptide was? Talked about a peptide bond. What's a peptide?
Short chains of amino acids.
Right, right. Honestly, almost any peptide is a polypeptide, which is a protein. So these are proteins that work as neurotransmitters. And they're like Rachel said, they're short. So they're stored in secretory vesicles and may also function as neuromodulators. Endorphins are the ones that, when you're working out, make you feel calm. They're the ones that can inhibit pain responses. My one experience that I can remember really well, I like to work out. I enjoy doing it. But at one point, and I don't do it now, I've learned that I have a curve in my lower spine, so I don't do it now. I used to run, and I used to enjoy it quite thoroughly. And I remember I was running up in a national forest and got back to my cabin and took my shoe off and realized that my foot was just soaked in blood, because I had cut one of my toes with the nail on one of my other toes while I was out running, and didn't recognize, because my endorphin level was so high at the time, I didn't recognize that I had been hurt until I finished and got home. But I just remember looking down at my shoe and realizing that I had been hurt and didn't recognize it at all. Neurotransmitters are excitatory, inhibitory, and sometimes the effect just depends on what type of receptor is found on the postsynaptic neuron. So the receptor is what's going to govern or dictate the effect of the neurotransmitter and its effect on the target cell. So acetylcholine is a common neurotransmitter that's released by many peripheral nervous system neurons. It's excitatory at the neuromuscular junctions and inhibitory at other synapses. So when it binds to a muscle, when the neuron synapses with a muscle, acetylcholine is always excitatory. However, at the other synapses, it's inhibitory. And there are multiple receptor types for each neurotransmitter. So how does this all work? With chemical synapses, how do they work? A nerve signal arrives at an axon terminal and opens. So an action potential, let's put it a different way here. An action potential arrives at the axon terminal. It opens up voltage-gated calcium channels. The calcium enters the terminal and triggers the exocytosis of the synaptic vesicles containing acetylcholine. So what calcium is the key for the release of the neurotransmitter into the synaptic cleft. Vesicles release their acetylcholine, and afterwards, empty vesicles cycle back into the cytoplasm to be refilled by acetylcholine. The acetylcholine, once it's released via exocytosis, is going to diffuse across the synapse and bind to ligand-gated channels, or aka receptors, on the postsynaptic neuron. Sodium enters by the postsynaptic neuron, and potassium is going to leave using the same gates. As sodium enters, remember that sodium is higher outside of the cell, potassium is higher inside of the cell. So as sodium enters the cell, because it's always moving from a high to a low, it's going to depolarize the plasma membrane, producing a local postsynaptic potential. Same as the local potentials we talked about last week. This postsynaptic potential is strong. If it's strong or persistent enough, it will open voltage-gated channels in the axon hillock of the postsynaptic neuron and induce an action potential. Questions? During the events involved in information transfer across a chemical synapse, which of the following steps would be directly interrupted by exposing a neuron to a calcium channel blocker?
I'm going to take a guess and say D.
D. Why do you say that?
Because the calcium channel is blocking the exposing neuron, or it's interrupting it.
How is it interrupting it?
By changing what the neuron is going to end up doing.
How does it do that?
because it reads the chemical or the hormone or the mineral, I guess, that's being attached to it.
Oh, you know what, Miranda, before you keep going, because you're kind of right. Where is calcium acting here?
In the synaptic vesicle.
In
No.
the synaptic vesicle, or the release of it from the presynaptic neuron?
The release of it.
Right. So come back and look at these answers again.
So would it be A?
It's A. Miranda, I'm not trying to pick on you there. I'm happy you're responding because you tell me, because you notice that nobody else is talking. And so I'm happy you're responding because it tells me what you're thinking. And I just wanted I wanted to walk you through getting to the right answer there.
No, thank you.
You're welcome. So drugs can modify the effects of neurotransmitters. There are multiple drugs out there that work via their effects on neurotransmitters. This is the reason that many drugs and substances are addictive, is for this same reason. So they can stimulate or inhibit the synthesis of a neurotransmitter. They can enhance or block the release of a neurotransmitter. They can activate or block the receptors of specific neurotransmitters. We have lots of drugs that mimic neurotransmitters in their structure. And for that reason, they can activate or block receptors. They can stimulate or inhibit the removal of a neurotransmitter from the synaptic cleft.
Can I ask one question?
Of course you can.
Because everyone swears up and down and around that weed isn't addictive. Can weed be addictive as a drug?
You know what? I don't know a lot of people who have. I know a few people who have done weed. I tend to run in a very what do you call it? Very I'm a little naive, Quentin, but one of my best friends who admitted to being addicted to weed, his thing to me was he gets addicted to the feeling of it. And I think if we start talking about other things, which our next slide is going to talk about how nicotine is addictive. Why do people smoke? Let me turn this around on you. Why do you think people smoke? Have you ever talked with a smoker and asked why they do it?
I do. Well, my mom used to. She said it was like a stress relief or whatever.
Uh-huh. So it calmed her down, right?
Yeah.
I think that is the same way that pot works, marijuana works. Now, does it work through the same process? Probably not. I don't know a lot about exactly how marijuana works. But I think people a lot of times get addicted to the feeling of it. And I'm guessing because most feelings are working through your neurotransmitters, that's a possible effect. Does that make sense?
Yeah.
So I would think, yes, from this one friend who told me he was addicted to it, he said that he got addicted to the feelings of it. And I don't know enough about it to know otherwise, but I could see how that could happen because I have friends who smoke who say they won't quit smoking because they like the way it makes them feel. So I think it really is the neurotransmitters, and we'll talk about it here with nicotine on this next slide. It really is all based on how they're affecting the neurotransmitter. What would be interesting is to look up, and bear with me just for a quick second, we'll divert because that's a really good question. I'm going to look up a paper right now. So this is my favorite medical article research. This is a federal government medical research database, www.pubmed.gov. I think that's what it is. And what I'm going to put in here is I'm going to put in what Quentin asked about. I think the major one that is addictive for people is THC, and I can't remember what it stands for. But I'm going to put it in there like that, and I'm going to say an addiction and see what happens. THC addiction. Search. Cannabis addiction and the review. This is an awesome article because this came out in 2018, and it's a free article, which means that I can open it up really easily. So I'm going to click on it here. Anytime if you're curious, if you look here on this site, anytime that it says it's a free article, that means that you can open up the full article. So cannabis addiction in the brain of a review. Quentin, I like this question. This is a good question. Thank you. I like what you were thinking about. Cannabis is the most commonly used substance of abuse in the United States after alcohol and tobacco. With a recent increase in the rates of cannabis use disorder and a decrease in the perceived risk of cannabis use, it is imperative to assess the addictive potential of cannabis. Here we evaluate cannabis use through a neurobiological model of addiction proposed by Kolb and Valkov. The model proposes that released substance abuse drives neurobiological changes in the brain that can be separated into 3 distinct stages, each of which perpetuates the cycle of addiction. So what this says is that the use of marijuana can result in neurobiological changes in the brain that allow for perpetual cycles of addiction. Let me say it differently. They can perpetuate the cycle of possible addiction. Here we review previous research on the acute and long-term effects of cannabis use on the brain and behavior. And find that the 3-stage framework applies to cannabis use disorder in a manner similar to drugs of abuse, albeit with some slight differences. What I'm going to do really quick is because I'm just curious now that Quentin got me thinking about it, I'm going to click on this. Quentin, it really is your fault, just so you know.
My apologies, everyone.
So we're going to click on this article. I want to see who did it. So it was the Journal of, and of course I can't read that last top line. Journal of, I can't read it. coming into it, what I want to see first is I want to see, I'm going to click Control F and see if I can pull this. Yep, let's see. I want to say neurotransmitter and see what happens here. So many different, so reading this, many different pharmacological treatments, so many different drugs have been investigated for reduction of cannabis withdrawal symptoms, primarily through the modulation of the receptor. So this is receptors, meaning that the cannabis binds to a receptor and activates a pathway. So what can we use if we're dealing with cannabis withdrawal symptoms? What can we use to bind to the receptor and allow a person to have less withdrawal symptoms from cannabis? You activate that receptor using something different.
Would it be CBD?
I think that the CBD is maybe you know what, I don't know enough about cannabis to tell you, but I think CBD isn't CBD the addictive part of cannabis?
I always thought it was THC because they sell CBD over the counter for like anxiety and stuff, and it doesn't give you the high that THC does.
Yeah, it's everything but the THC to my extent of knowledge. So you don't like get high taking CBD, which is like why it's able to be sold over the counter.
And they say it won't come up in a drug test either.
Yeah.
Really? Okay.
As long as you got to be using a lot of CBD for it to be coming up in a drug test, because there's always what they call trace THC. So that's why you got to really watch what you buy over the counter and make sure that you're getting what you're actually paying for with CBD products, because gas stations sell them over the counter and everything.
Right, right. I don't know. So it says through other neurotransmitter systems, including the glutamate, dopamine, norepinephrine, serotonin, and GABA. In their comprehensive review of the pharmacological treatments for cannabis use disorder and cannabis withdrawal, so these people conclude that therapies targeting specific symptoms of withdrawal should be administered in conjunction with the target reduction of cannabis use. Some promising candidates. So these would be things that would be work to block the receptors and allow people to be less, hopefully less addictive than cannabis. So Quentin, a very long answer to your simple question is, in this paper, they say that yes, it can be addictive. And what they're trying, and it works through the neurotransmitter pathways. My guess is looking here, and when we go back to our PowerPoint here, that it's working through the dopamine pathway. One of the possibilities is it's working through so my guess is that it's working through a dopamine pathway. Let me do something else really quick before I keep going down this direction. I want to do this. I want to say THC and dopamine. the effects of THC on the dopamine system. So the effects of THC, the main psychoactive ingredient in cannabis, are pressing concern. I'm going to skip through some of this. Many of the reinforcing effects of THC are mediated by the dopamine system. Dopamine is the happy, it's the mood stabilizing. It's a mood stabilizing neurotransmitter. So owing to the complexity of the cannabinoid dopamine interactions that take place, there's conflicting evidence from human and animal studies concerning the effects of THC on the dopamine system. Acute THC administration, so giving people a lot of THC, causes increased dopamine release. So what we can say with this paper alone, I mean, I would have to read the whole paper, but what we can say from this paper alone is that it's that taking more THC in is resulting in more dopamine neurotransmitter release. The acute, so this is acute meaning not long-term. However, long-term use of it, so the acute meaning short-term. Okay, so let me rephrase this because I've never read this article. So I'm summarizing it for you and then I'm telling you what I think I know about it. What I think it's saying. Acute THC meaning short-term. Short-term THC administration causes increased dopamine release. However, the long-term use of it, so regular and consistent use of THC, will blunt the dopamine system. Future research needs to be taken into the long-term and developmental effects of dopamine effects of THC. What that means is that it initially using THC is going to result in increased dopamine release. Long-term wise, it's actually going to blunt dopamine release, causing you to have reduced dopamine secretion. So the whole reason at this point, Quentin, reading these papers, I would tell you, yes, cannabis or THC specifically is addictive.
Okay, that's what I thought, but everyone, like all my friends that are into that stuff are like, it's not addictive. I can stop when I want. I'm like, okay. And
well,
I can start thinking.
So sometimes this is why knowledge is power. The
Right.
more you learn, the more you understand, and the knowledge is power.
Yeah, and I tell them, I'm like, okay, you can stop smoking anytime you want, just like I can stop drinking caffeine anytime I want. It's not happening.
And that's kind of how I feel. I'll just say when it comes to addictions, I don't have a foot to stand on. I'm not a big fan of coffee. Like I've never been able to handle the taste of it. But I wake up with an energy drink in my hand.
Oh yes, something has to give.
Yes, exactly. So on this same response, and the idea here is nicotine, and we'll talk about nicotinic receptors later. But nicotine works on acetylcholine receptors. This is talking about cannabis. Nicotine itself is one of the most potent addictive substances. It's next to heroin in its potency for addiction, because it binds to acetylcholine receptors and increases dopamine release. And if I remember right, dopamine is your mood stabilizing thing. So this is what, even though it's activating neurons, it's calming people down. Even though it's bringing up their heart rate and constricting their blood vessels and reducing blood flow, it's calming people down. I'm sure none of you are probably, after that long discussion, and Quentin, I really like that discussion, because that's right now, that's a very big deal. It's if you want to learn, if you got that site, that's open to, you saw that how easily I activated and how easily I found it. That's open to the public. And so if you know, knowledge is power. So in contrast to excitatory responses through acetylcholine, inhibitory GABAergic synapses are synapses that employ gamma aminobutyric acid. So this is going to inhibit. What if it's excitatory? Does it hyperpolarize or depolarize the membrane? Someone tell me, what's the first part of an action potential initiation? Is it depolarization or hyperpolarization? All right, I'm going to call on somebody. Jessica, is it depolarization or hyperpolarization?
What was the question? I'm sorry.
Well, when an action potential is initiated, is it depolarization or hyperpolarization first?
Depolarization?
It is depolarization. So depolarization is going to result in an action potential being initiated. Hyperpolarization is what occurs when a GABA receptor opens a chloride channel. So the result is that this gamma aminobutyric acid is going to inhibit or reduce the possibility of an action potential being fired, because it's going to hyperpolarize the membrane, making it more, the resting membrane potential more negative. So rather than having being minus 70, it's going to be like minus 72. And remember that we have to reach, I think, was it minus 55 for an action potential to actually be initiated. So the result of GABA, this gamma aminobutyric acid, GABA, binding to a receptor is that it's going to hyperpolarize the membrane, making it less likely to fire an action potential. This is why it's an inhibitory synapse. These are both ion channels. So this acetylcholine channel is an ion channel. This GABA is an ion channel. Adrenergic, or working with norepinephrine, is going to work through a cyclic AMP mediated channel or a second messenger system. So it's not going to work through an ion channel. So norepinephrine or noradrenaline or other amines and neuropeptides are going to act via a second messenger system, which includes cyclic AMP. So these are G protein coupled receptors instead of ion channels. The neurotransmitter binds to a receptor. It activates a G protein. The G protein then activates an adenylate cyclase. The adenylate cyclase is going to bind to, the adenylate cyclase is going to activate the synthesis of cyclic AMP from adenosine triphosphate. So G protein activates adenylate cyclase. Adenylate cyclase results in the synthesis of cyclic AMP. And cyclic AMP goes on to have several other effects. Those other effects is that it can produce an internal chemical that will open a ligand-gated channel and allow more ions to flow into the cell. It can activate enzymes that are going to lead to a variety of metabolic other changes, or it can induce the transcription of genes encoding for enzymes involved in other metabolic activities. So what you want to remember from this is that neurotransmitters can work through ion channels, or they can work through second messenger systems. Even though these second messenger system activation is a slower process, there's a lot more pathways that have to be moved through. These work by signal amplification. So they end up activating multiple signaling cascades. One neurotransmitter can result in the formation of many cyclic AMP molecules and induce the transcription to generate many mRNA molecules, resulting in the production of many enzymes. Questions? Well, we've got to be able to get rid of a neurotransmitter. Once we release it, eventually we need that neuron to stop firing. So how do we go about doing that? We've got to remove it from the synaptic cleft. It's going to keep binding to the receptor the longer it's there. and it's going to keep activating things when it's there. So we've got to get rid of it. What the 3 ways that we can get rid of it is we can degrade it. An enzyme in the synaptic cleft breaks it down. Acetylcholine esterase breaks down acetylcholine. We can reuptake it, we can reabsorb it. So a neurotransmitter, the broken down fragments, are reabsorbed by transport proteins in the axon terminal. Once the amino acid and monoamine transmitters are taken up, they can be broken down by what's called a monoamine oxidase. And then they get reused. They get recycled and put back into synaptic vesicles for the next time. One other way that the signal can go away, the neurotransmitter can leave, is it can diffuse away into the extracellular fluid. So it gets broken down, gets retaken up by the presynaptic neuron, or it diffuses away. Questions? A very popular drug, Prozac, aka fluoxetine, works by, I have to remember this. It works by blocking the reuptake of serotonin. So serotonin is your happy stimulating neurotransmitter. And oftentimes in people who struggle with depression or anxiety, Prozac works on depression and anxiety levels. It works through people who struggle with anxiety and depression often have reduced serotonin stimulation. They either have reduced release from the neurotransmitters, or their receptors, there's not enough receptors for it on the postsynaptic cell. So one way to keep more serotonin around for people who struggle with depression and anxiety is to block how much it's being reuptaken into the cell, because that keeps them in the synaptic cleft longer. So when it's released, it stays in the synaptic cleft longer. And therefore it can stimulate more neurons for a longer period of time, because it's not taken up as easily. Questions? You guys, let's take 10 minutes. I need to get up and move around. I'm sure you do too. Let's take 10 minutes right here. So we'll come back at 10:49. And we will finish this chapter and start chapter 13. Miranda, you here?
Yes.
I just saw your email just now. Why, if you did those, send me screenshots showing you completed them.
Can I share my screen with you right now?
Actually, can you do it? Can you send me screenshots?
Yeah.
Because what I'll do is I'll update your grades after class.
Well, the second two that I was referring to, I can't find them. Like, I don't know what week they were even in for me to miss them, to be like, did I truly miss them or not?
We did them during the connective tissue. They were virtual labs during the connective tissue, or during, sorry, not connective tissue. Lab exercise 6.
Yeah, because lab exercise 8 and 9 is when I crashed my car. So I think I really did truly miss them, but I just don't see them. So lab exercise 6, do you remember what week that was by chance?
I can look really quick. Give me 1 second. Let's see here. Give me 1 moment here.
C and D?
Say it again.
Was it C and D? Oh, I see.
Yeah, it was. Yeah, it was.
Okay.
If you can't open them, let me know, and I will push back the deadline, and you can get in there and do it. Okay.
Well, it says the deadline on.
They're not due yet.
No, wait, C and D. We're talking about. Let
me show you. So give me 1 second here. Okay, not that one.
Week of.
Here. So they were right here.
Okay, yeah, mine say the same due dates, the 12th and the 26th.
Yes.
Okay, then I don't know why they have zeros in already, but.
Well, because you know what, I don't know. I should have changed those due dates and I didn't. But just get them done.
Okay, no problem.
You know, they're gonna have zeros until they're done. But that doesn't mean that they're being added into your grade. Does that make sense?
Yeah, well, most of my other grades that like we haven't done yet have dashes, and that's why I was confused. because I was like, oh my God, did I miss these 2 assignments? Like, I have no clue what they are, to even be like, did I do them and check to see? Because there were other assignments that I was like, oh no, I truly did or didn't miss this. Like, I wasn't going to harass you about that kind of stuff when I can
You
look for
can
my
you can harass me about that kind of stuff. Don't. I may, like you and I both learned, I may not see your email. But you can harass me about anything you want to. Okay?
Yeah, thank you.
Welcome.
9.7.
I don't take offense to that kind of stuff.
I just know people have time to themselves too.
Yeah. Well, no, you know what, right now, my focus during the semester is my classes. It's a marathon for me. I told you I like to work out. That's what I do. Do that once a week just to center myself a little bit. But outside of that, this is my life. So don't worry about it.
Okay. Thank you. Oh, I did want to ask.
Yeah.
There are like 6 assignments due on the 10th. Is that true?
6 or so, say it again.
There are like 6 different assignments that are due on the 10th of November. And I just didn't know if that was true or not because I missed that last.
What ones are they?
It says according to the calendar, like the one that shows you the date and like it actually shows you the assignment due. There's the nervous system, monosynaptic reflexes. There's chapter 12 nervous tissue.
So the monosynaptic reflexes, yes, they are probably all due then.
Okay.
But they've been open for a while.
Okay.
That is about when I think the next exam will be.
Okay, because there's also like 3 labs due that day too, so I didn't know.
What labs were they?
16, 17, and 18.
Yes, because see, that's everything due. We're going to do lab 16, 17, and 18 in lab this week. Does that make sense? So they are due the following Wednesday. Does that make sense?
Yes.
Okay.
Hi, this is Asanti. I was wondering if you received my email.
I did. I just looked at it. You know what? How are you missing scores?
It has like slashes, but the score, like, they weren't due yet.
Yeah, but you know what? I'm not going to open your grades right here because everyone in the class can see, and that's a major FERPA violation. If go back and make sure that things that were due you've done. So everything through chapter 7 has already is past due.
Yeah, I did everything but like one thing that was like worth 10 points, but that's it.
Have you checked your grades? Are they all there?
Yeah, like when I calculate the little number up top, it's like a 90 something percent.
I would have to look because I can't tell you right now. Okay. Is it still that low? You're not going to tell everyone what your grade is, but is it still that low right now, or was that just midterm?
It was a midterm grade that was really low. That's why I was like,
change now. It can change now. If you did that stuff since then, it's going to change. Because midterms were about 2 weeks ago.
Okay.
So look at it now. Look at what your grade is now. Nobody cares what your midterm looks like at the end of the semester. It doesn't get put on your permanent record. It does not, all it is it's a heads up saying this is where you are at this moment in the semester. That's all that it means. Look at your grades now and see if it's still that low. And if it's still that low, then we should talk about it. Does that make sense, Asante?
Yeah, I just thought I completed everything by the due dates. That's why I was so confused why it was low.
I don't know. I can't tell you, because I can tell you I remember very little from 2 weeks ago.
Okay.
Let me continue on here. So neuromodulators are molecules that are secreted by neurons with long-term effect on entire groups of neurons, rather than brief, quick effects of neurotransmitters. They are going to be involved in the increased release of neurotransmitters by presynaptic neurons. They can adjust the sensitivity of postsynaptic neurons to a neurotransmitter. So they can hyperpolarize a membrane so that when an excitatory response comes in, it doesn't have as big of an effect. And we'll talk about that in a few minutes here. They can alter the rate of a neurotransmitter reuptake or breakdown. So they can alter how long is it here in the synaptic cleft. Nitric oxide diffuses into a postsynaptic cell and activates second messenger systems. It can relax smooth muscle to dilate small arteries and increase blood flow. Neuropeptides such as enkephalins and endorphins inhibit spinal neurons from transmitting pain signals to the brain. This is why I told you about that story of being out running and coming home and finding my sock soaked with blood. It inhibits spinal neurons from transmitting pain signals. Other hormones and the neurotransmitters dopamine, serotonin, other neuromodulators are dopamine, serotonin, and histamine. So one thing that's very important to understand when we talk about neurotransmitters, neurohormones, or neuromodulators is a given substance can work as all three. Why we call it a neurotransmitter, why we call it a hormone or a neuromodulator is based on the role it plays in a given situation. One chemical can play two or more roles depending on the situation. So lots of times we don't classify given substances as being neurotransmitters, hormones, or neuromodulators unless we're talking about a specific situation, because they can function as all three. Any given substance can function as all three. I hope this gives you a new respect for how much goes on in the nervous system. The more you learn about the nervous system, the more you learn what we don't know about it and the complexity of it. So neural integration, you remember we talked at the very beginning today, we talked about synapses having the one synapse in the cerebellum, one neuron in the cerebellum can synapse with up to 100,000 different presynaptic neurons. So the more synapses a given neuron has, the greater its information processing capabilities. Sometimes, just like we talking about Quentin when we were talking about the cannabis addiction, more knowledge is power. Same thing for the synapses. The more information available, the more one can make a decision and make a confident decision. A pyramidal cell in the cerebral cortex has about 40,000 synapses with other neurons. We'll talk about the cerebral cortex in chapter 14. Neural integration is a neuron's ability to process information, store and recall it, and make decisions. And this is all based on postsynaptic potentials that are initiated in the axon's trigger zone. We have excitatory postsynaptic potentials, and we have inhibitory postsynaptic potentials. Excitatory postsynaptic potentials is where there's a voltage change that makes the neuron more likely to initiate an action potential. This meaning that the more the neuron, the resting membrane potential or the membrane potential becomes more dipolar, becomes more positive. This is an excitatory postsynaptic potential. An inhibitory postsynaptic potential is the hyperpolarization, making the membrane potential more negative than it was before. Making the neuron much less likely to fire an action potential. Excitatory postsynaptic potentials occur because sodium flows into the cell. Inhibitory postsynaptic potentials are because chlorine is going to flow into the cell, or potassium is going to flow out. So excitatory, meaning that where the membrane potential becomes more positive, meaning it gets closer to the threshold. Inhibitory postsynaptic potential, meaning the membrane potential becomes more negative, gets closer to, it gets further away from threshold. Remember what happens at threshold, that's where we fire an action potential. So excitatory postsynaptic potentials and inhibitory postsynaptic potentials don't dictate whether a neuron will fire an action potential. They just change the rate of firing by stimulating or inhibiting the production of more action potentials. So potassium flowing out of a cell is going to result in the cell becoming more, the membrane potential becoming more negative, meaning a hyperpolarization. So it's an inhibitory postsynaptic potential. Chlorine, which is negatively charged, flowing into the cell is going to make the membrane potential more negative, meaning that it's hyperpolarizing or inhibitory postsynaptic potential. Sodium, which is positively charged, flowing into the cell is going to make the inside of the membrane potential more positive, meaning it's going to be an excitatory postsynaptic potential. So normally a given excitatory postsynaptic potential cannot initiate a nerve impulse. Summation is the process of adding up postsynaptic potentials and responding to their net effect. So we got one given postsynaptic neuron receives multiple signals, some positive, some negative. The more positive they are, the more likely we are to reach threshold. However, we can receive signals that cancel out the positives. So ultimately, the whole goal is, do we reach threshold at the axon hillock or do we not? That's summation. Let me give you something, way to think about this. So let's say that I have to reach a zero. We'll say I have to reach a negative, let's see. I have to reach a negative 55 to reach. So I start with negative 55. Nope, I'm sorry. I start with negative 70. In order to reach threshold, I have to reach a negative 55. So I get a negative 2 first. So now I'm at a negative 72, which means that I'm less likely to reach threshold, right? Because now I'm at a negative 72. Then I get a positive 10. So negative 72 plus 10 puts me at a negative 62. So I'm more likely to reach threshold, right? But then I get a negative 5. So now I'm at a negative 67. However, then I get a positive 2 and a positive 3. So now I'm back to negative 62 again. Now I'm at negative 62. Well, now I get a positive 7. So I was at a negative 62. Now I get a positive 7. I've hit threshold and I fire an action potential. Does this make sense how this summation works? Somebody tell me yes or no.
Yes.
I mean, kinda. I'm just not good with like math and stuff like that.
Okay, just, you know, what we're doing is we're just adding up positive and negative numbers. Just keep in mind that if you, the more negative you get, the less likely you are to reach this threshold value, right? If you start at negative 70, the farther you are away from threshold, the less likely you are to fire an action potential, because you have to reach threshold to fire an action potential. All of these little ones right here that we're getting, these are all local potentials until we reach this value. So all of these are local. And so the more negative it becomes, the less likely we are to reach this.
So the threshold is negative 55 always?
Always. It depends on the cell, but in the case of the neuron, it's negative 55. When we talk about muscle at the very end of the semester, we'll learn that that's not the case in muscle. It's different in muscle. But right now, it's all, threshold is a certain number that you have to reach. The more negative you get, the less likely you are to reach that value. The more positive you get, the more likely you are to reach that value. But things are constantly being summed together until you reach threshold, and then it's an all or nothing at that point. It all occurs at that point. And think about it for a little bit, but keep in mind that it's a value, it's adding up negative and positive numbers until you reach negative 55. And then it fires. But the more negative it becomes, the less likely you are to reach threshold. The more positive it becomes, the more likely you are to reach that threshold value. So this is gonna, the summation where we reach threshold is going to occur in the trigger zone. Temporal summation is when one presynaptic neuron is generating so many excitatory postsynaptic potentials on one postsynaptic neuron that each potential is generated before the last one has faded. They're happening so frequently that each one is occurring before. Remember how local potentials are decremental, so they decrease over time. Well, if one presynaptic neuron is firing so many at one time, it's going to keep firing them, and one is going to be added onto the next one because the second one comes in before the effects of the first one are gone. So all of these excitatory postsynaptic potentials from one neuron are reaching the threshold. Add up to the add up to the voltage required to reach threshold at the axon hillock. Spatial summation is when they're coming from when the excitatory postsynaptic potentials are coming from multiple neurons. Onto one postsynaptic neuron. So this results, so the adding up of multiple excitatory postsynaptic potentials from different presynaptic neurons are going to add up. Temporal is when they're all coming from one presynaptic neuron and they're coming so quickly that the effects of one adds on to the effects of the last one. Suppose the excitatory postsynaptic potentials summate in a postsynaptic neuron in response to simultaneous stimulation by neurotransmitters glutamate, serotonin, and acetylcholine, released by 3 separate presynaptic potentials. Presynaptic neurons. Is this an example of spatial or temporal summation? Say it again. What's the key word here in this sentence? Temporal is coming from one presynaptic neuron. Spatial is coming from multiple ones.
So would it be spatial?
It's spatial, right? Which of the following is true, a true statement about neurons?
I think it's C.
I think you're right. I know you're right. Remember, we have these absolute refractory periods. Things can only move in one, potentials can only move in one direction. So that's the reason dendrites conduct signals from the cell body. Away from the cell body. Wait. Nope, it's not C. It's not C.
No, I was either mixing them up, so then it has to be B.
It's B. So they conduct them towards the cell body because dendrites, remember, those are the feelers on the cell body. So they're conducting it towards the cell body. Axons are conducting signals away from the cell body towards the axon terminal. A membrane depolarization is caused by what? Membrane depolarization is caused by what?
Is
Is it A?
It's A. So it's sodium ions. Which of the following is not true about synapses? Guys, it's C. Because neurotransmitters, do they bind on both sides of the synapse? Do they bind to the presynaptic and the postsynaptic neuron? No, they only bind to the postsynaptic neuron. So that's where they cause ion channels to open. Sodium potassium pumps help maintain membrane polarization by what? Guys, what do the sodium potassium pumps do?
Is it D?
Yes.
What was the last answer again?
Actually, wait. Think of, you're close. It's not D. What else could it be? Does it pump sodium into the cell or does it pump it out?
Pumps it out, so it's E.
It's E. What was your, who just asked a question?
Me, I did. I missed the last answer.
Okay, so this one, neurotransmitters, they only bind to the postsynaptic neuron. So they can't, this one, not sure about synapses. Neurotransmitters bind to receptors on both sides of the synapse. No, they only bind to one. They bind to the postsynaptic neuron.
Okay, thank you.
Okay, last part of this chapter, presynaptic facilitation. So one synaptic neuron can enhance the effects of another presynaptic neuron. One facilitating neuron releases a neurotransmitter which allows for calcium channels to stay open longer in another for that neuron. The result is that the neuron R, the responsive neuron, is excited for a longer period. So one neuron, neuron S, results in the firing of an action potential. Neuron F results at once that action potential reaches the bottom of the cell here, the axon terminal. The facilitating neuron is going to initiate more activity, is going to cause the calcium channels to stay open longer, resulting in this neuron firing longer, resulting in this neuron being activated for a longer period. So neuron F is enhancing the abilities of this neuron. Which allows for the neuron, the postsynaptic neuron, to be excited for a longer period of time. That's presynaptic facilitation. Opposite is presynaptic inhibition, where one presynaptic neuron is going to inhibit the effects of another presynaptic neuron. Neuron I releases an inhibitory neurotransmitter which prevents calcium channels from opening or slows down how long they're open, keeps them closed, causes them to close prematurely. How about we don't say causes them to close prematurely? How about we say causes them to close faster? And the result is that neuron S is going to release less neurotransmitter. and neuron R will either be not activated at all, or it will be activated for a much shorter period. Questions?
No.
Neural coding is the mode by which information about and around the body is communicated in a meaningful pattern of action potentials. Call this the labeled line code. The basis is that each nerve fiber receives from a receptor and recognizes only one specific stimulus. One receptor receives one kind of stimulus. Now, this is an odd one, and it's always fun to do because we've all done it at some point. And I'm not wearing eye makeup, so I can do this. You look, your eyes, the receptors in your eyes, they are looking at one type of light. Not one type of light. They only respond to light. They're receptors that are only initiated and activated by light, right? So keep that in mind. How many of you, especially when you have a headache, you put your hands up against your eyes and you press on your eyeballs?
Me all the time.
Okay, my question for you, Quentin, is do it right now. What do you see when you do that? When you push on your eyeballs and you try to see, what do you see?
It's like kind of reminds me of like colors. I keep seeing colors, but
you see like colors and maybe some black spots.
Yeah.
What that is your receptors are trying, the photoreceptors in your eyes are trying to take that pressure that's coming from that pressure sense of pressure on your eyes. Your photoreceptors that only know how to respond to light are trying to make sense of the pressure stimulus. And so what you see are that light and dark splotches, because the photoreceptors in your eyes can only respond to light. And so they're trying to make sense of it. And they can't. So any stimulus that's received from the optic nerve is going to be interpreted as a form of light, whether it's light or pressure. A little bizarre, huh? You probably never thought of it that way. I can say that I hadn't until I read that in your textbook. I thought it was pretty awesome. So a stimulus strength is interpreted by how many neurons are firing. Different neurons have different thresholds of excitation. A weak stimulus excites neurons with the weakest threshold. A strong stimuli is going to excite neurons with higher thresholds. Additional neurons will be recruited as the stimulus becomes stronger. A stimulus intensity is judged by the firing frequency. How often is the stimulus coming in? A weak stimulus can cause a neuron to generate 6 action potentials per second, while a strong stimulus can generate 600 per second. Questions?
No.
Neurons function in larger assemblies called neural pools. Each pool can consist of thousands of interneurons. Remember the interneurons, the central nervous system. Each pool can consist of thousands of interneurons concerned with a particular function. Neural circuits are a complicated network of neurons that process specific types of information. Such a stimulus, actually, let me start. So a diverging circuit. So different circuits, complicated networks. So we have different networks of neurons. A diverging circuit, one nerve fiber branches and synapses with several postsynaptic nerve cells. Each one may then go on to synapse with several more. This type of circuit allows one motor neuron of the brain to stimulate thousands of muscle fibers. A converging circuit is input from many nerve fibers that's funneled into one postsynaptic neuron. This allows input from your eyes, inner ears, stretch receptors in your neck to be directed to an area of the brain concerned with the sense of balance. A reverberating circuit is where neurons stimulate each other in a linear sequence from input to output neurons. But some neurons late in the path send axon collaterals. So you remember axon collaterals are ones that can come back and can synapse with the axon. Axons that can come back and synapse with previous axons. So neurons stimulate each other in a linear sequence where some neurons late in the path send collaterals back to neurons earlier in the path and re-stimulate them. This allows for circuits to send repetitious signals to your diaphragm and intercostal muscles to make you inhale air. When the circuit stops firing, you exhale, and the next time it fires, you inhale again. A parallel after discharge circuit is where a single presynaptic cell stimulates a group of neurons. And then they synapse with a common postsynaptic cell. So continued firing after the initial stimulus stops is called after discharge. Here. Explains why you can stare at a lamp, close your eyes, and continue to see an image of it for a while. Questions? All right. Okay, this is the end of chapter 12. Let's start chapter 13. Oh, what did I do? Give me a second. This was in the middle of it. Come back. No. We will finish a good part of this today. So the spinal cord, the spinal nerves, and somatic reflexes. You guys, what's the somatic nervous system? What did the somatic nervous system entail? Someone tell me. What does the somatic nervous system entail?
It includes all the nerves that run to and from the spinal cord.
So, yep, it includes all of the skeletal muscles, joints, and skin and bones. So the somatic reflexes are going to have to do with skin, joints, bones, and muscles, skeletal muscles. Spinal cord injuries in the US. The spinal cord is the information highway that connects the brain with the lower body. However, thousands of Americans are paralyzed by spinal cord injury every year with devastating effects on their quality of life. Why do you think this is the case?
Cells don't regenerate.
Yeah, they don't regenerate. Neurons don't regenerate. Once they're out of the game, most of the time they're out of the game. Anything below a certain level, if you lose connection between the brain and the spinal cord, anything below where that break occurred or wherever the cut occurred, everything below it is lost. Everything that was innervated by the spinal cord below that cut, the functions are gone. So different functions of the spinal cord. The spinal cord is involved in conduction. So nerve fibers that conduct sensory and motor information up and down the spinal cord. Neural integration. The spinal neurons are going to receive input from multiple sources and integrate it and execute appropriate output. So just like we saw in the very last part of chapter 12. Locomotion. The spinal cord contains central pattern generators. There are groups of neurons that coordinate repetitive sequences like the contractions for walking or going up and down stairs. Reflexes, involuntary responses to stimuli that are vital to posture, coordination, and protection. How many of you curl your hair with a curling iron?
On occasion.
On occasion, Erin. Have you ever grabbed the wrong end of the curling iron?
No, but I've definitely like got my ear and my neck with the curling iron.
Okay. At some point, you need to grab the wrong end of the curling iron. You're living kind of a sheltered life there. Someone, I thought it was Miranda, said before that she watched it blister. I think
I watched, like, I just looked at my hand because I grabbed it with my left hand and I wasn't used to using my left hand. So I like looked at it for a good 10 seconds before I dropped it. And then I could see it blister on my hand.
Ouch.
How,
I think I'll let the other people live dangerously.
Most people outside of Miranda, when she grabbed it with her left hand, most people drop that before they even realize it's hot. That's a somatic reflex. So it's an involuntary stimulus. You don't have to think. Most of us, outside of Miranda, most of us don't have to think about dropping it. We just do it. The spinal cord is a cylinder of nervous tissue that arises from the brain stem at the foramen magnum of the skull.
I think your screen is frozen. All I can see is the first slide.
Really? Let's try this again. What about now?
Yeah, I can see it now.
Okay. So I just talked about conduction, neural integration, locomotion, and reflexes. Spinal cord is a cylinder of nervous tissue that arises from the brainstem at the foramen magnum. It contains about 100 million neurons, with about 50 times more neuroglia. Remember when we talked about neurons versus neuroglia, that there were about 50 neuroglia for every one neuron? The spinal cord extends from the medulla oblongata, here right next to the brain, to the conus medullaris. Spinal cord injuries commonly result from fractures of the C5 to C6 vertebrae, but never of the L3 to L5 vertebrae. Why?
Is it because those ones are kind of like tucked under your butt a little bit?
I would think so. I would think so. And your neck is so much more vulnerable. If you think about all the flexibility of your neck, do you have that kind of flexibility in your back? I mean, like I can roll my head all the way back. Can I do that with my back? No. I would think that's why. Is because and the vertebrae are also much smaller. Also, probably a good reason why you have the flexibility you have of your neck. The spinal cord occupies the upper 2/3 of the vertebral canal. It only extends to about L1. You remember we talked about the vertebral column. You have the cervical vertebrae, the thoracic vertebrae, the lumbar vertebrae, and the sacral vertebrae. The spinal cord, it only extends to about L1 in adults. It gives rise to about 31 pairs of spinal nerves. Each spinal cord, part of the spinal cord, is occupied by a pair of spinal nerves called a segment. So you have the cervical segment, the thoracic segment, the lumbar segment, and the sacral segment. It's two major enlargements. The cervical enlargement is going to give rise to the nerves of the upper limb. The lumbosacral enlargement is going to give rise to the nerves of the pelvic region and lower limbs. And the cauda equina is going to give rise to a bundle of nerve roots that occupy the vertebral canal from L2 to S5. L2, lumbar 2 vertebrae, to the 5th sacral vertebrae. Any of you horse people? Anyone really like horses?
They're okay.
They're okay. My mom, if she was here, she's a big horse person. What does equine mean? Equine means a horse. Cauda meaning a tail. So this kind of looks like a horse's tail. The spinal nerves are divided into 4 regions, named for the level of the vertebral column with which those nerves arise. The cervical spinal nerves, the thoracic spinal nerves, the lumbar spinal nerves, and the sacral spinal nerves. There's 8 pairs of cervical nerves. You guys, how many cervical vertebrae are there?
7.
There's 7. So keep that in mind for a second here. There's 8 cervical nerves that control the sensory and motor activity of the neck, head, neck, upper body, arms, and hands. The thoracic nerves have 12 pairs. They control the sensory and motor activities of the hands, fingers, chest, and abdominal muscles. Lumbar nerves control the sensory and motor activity of the hips, knees, ankles, and toe muscles. There's 5 pairs of sacral nerves that control the sensory motor activity of the legs, toes, bladder, and anal muscles, and 1 coccygeal nerve. How many thoracic vertebrae were there?
12.
How many lumbar vertebrae were there?
5.
How many sacral vertebrae were there?
5.
And so my question is for you, for the thoracic, lumbar, and sacral vertebrae, we each have 12 vertebrae, 12 nerves, 5 vertebrae, 5 nerve pairs. Sacral nerves, 5 vertebrae, 5 nerves. How do we get 8 up here? The reason for this is there's 1 coming above the first cervical vertebrae. All of the others are going to come underneath it, the vertebrae. There's 1 coming above in the cervical. So there's 1 coming above C1. So each of the others, and I don't have a good picture of it. Let me show it to you really quick here. Looking at put away my little mini skeleton last night, or this would have been a good time to show him again. Intervertebral foramen and spinal nerve. What's a foramen? Do you remember?
A hole.
It's a hole. So here's the intervertebral foramen.
We can only see the PowerPoint.
Oh, shoot. Let's see, stop sharing. Thank you for saying something. Here's the intervertebral foramen. Here's the spinal nerve coming out. So that's the way it's going to come out. Here's, let's say, for example, and I don't know if it tells us which, no, it doesn't. Let's say that this is L1. This is the L1 spinal nerve. Lumbar vertebrae 1, lumbar nerve 1. So this, I mean, we talked about how an injury to the spinal cord is pretty devastating. Because there's not a whole lot you can do because they don't undergo mitosis, neurons. There's a lot of protective layers involved in protecting the nervous tissue. The vertebrae provide a strong connective defense against damaging blows and bumps. There's a lot of bone encased here. An intervertebral disc that keeps things cushioned and keeps spinal nerves from being pinched. When these discs become compressed or herniated, the discs start to get pinched. Meninges are another level of protection.
I think your screen is frozen again.
Ah, really? This is going to get a little old. Let's see here. What about now?
Yep, that's good.
Okay, thank you for giving me the feedback here. So 3 protective meninges. These are fibrous connective tissue. So this is dense, irregular connective tissue that encloses both the brain and the spinal cord. So these are like sheets that are going to completely cover the brain and the spinal cord. From superficial to deep, so from the most outer layer to the most inner layer, the mnemonic for remembering them is DAP. Dura mater is the most outer layer. It surrounds the spinal cord and it's separated from the vertebrae by an epidural space. If you've had a child or you know someone who's had a child, what do a lot of them have done before they have the child? Probably have a lot of things done, but what's something they do in the hospital to help with pain?
Epidural.
Epidural. What they do is they inject it into the epidural space. That's why it's called an epidural. So epi meaning on top. So this is a layer, a space between above the dura mater, in between the vertebrae and the dura mater. Arachnoid mater adheres to the dura and is separated from the pia by fibers spanning the subarachnoid space, which is filled with cerebrospinal fluid or CSF. I am a chicken when it comes to scary movies. I don't like scary movies. I learned really early in my lifetime that my imagination is really pretty amazing and that scary movies are not good for that reason. Things become way too real for me. One of the most scary movies that I ever saw in my childhood, or in give you a guess as to how old I am, I was my adolescent period. I saw the movie Arachnophobia. What does that mean? What does it mean to have an arachnophobia?
Isn't that the spider?
It's the fear of spiders. If you remember, there was a really old movie that was called Arachnophobia. If you don't like spiders, don't watch that movie. What arachnoid means is that this membrane, this arachnoid mater, looks like a spider web. It's a pattern of webbing. And the subarachnoid space means the space right underneath the subarachnoid mater. So the three basic meninges are the dura mater, the arachnoid mater, and the pia mater. The epidural space is above, right superficially to the dura mater. The subarachnoid space is right underneath the arachnoid mater. And the pia mater is a delicate membrane of 1 to 2 layers of squamous or cuboidal cells that supplies blood vessels with nutrients and nervous tissue with nutrients. It's see-through. If it's 1 to 2 layers of squamous or cuboidal cells, it's see-through. When you look at a brain or a spinal cord, you're looking through the pia mater. You're probably sloughing it off before you even realize what you're looking at, because it's that thin. The dura mater is exceptionally tough. Dura mater means hard mother or tough mother. It's exceptionally tough material. Arachnoid is the spider webby like material. Pia is very soft. You're going to see through it when you're looking at the surface of the brain or spinal cord. Denticulate ligaments are the thickenings of the pia mater that are going to suspend the spinal cord in the dura mater sheath. Little tiny thickenings or thickenings, little extensions of the pia mater. The epidural space contains blood vessels, adipose tissue, and loose connective tissue. The subarachnoid space contains circulating cerebrospinal fluid, which is going to cushion the spine and deliver nutrients and remove waste. So here's the dura mater right here. Here's the epidural space. Here's the subarachnoid mater. Here's the pia mater. right in between the pia mater and the arachnoid mater is the subarachnoid space. So looking here, here's the epidural space. This blue line right here is the subarachnoid space. Talk about more when we talk about the brain, the significance of the cerebrospinal fluid, but recognize that it's throughout the subarachnoid space in the brain and spinal cord. So these meninges are continuous sheaths, and the subarachnoid space is where the cerebrospinal fluid circulates around the brain and the spinal cord. A lumbar puncture or spinal tap is a sampling of the cerebrospinal fluid in the subarachnoid space to look for bacteria, blood, white blood cells, or other abnormalities. This is what they do to diagnose meningitis or other diseases. Meningitis meaning inflammation of the meninges. Agents such as drugs, chemotherapeutic agents, analgesics, or contrast media can also be injected into the subarachnoid space. Have you ever known someone who's gotten a spinal tap or a lumbar puncture? I haven't had it done either. But this is cerebrospinal fluid is fluid that's coming from the blood. It's secreted from the blood into the subarachnoid space. And when people get a lumbar puncture or spinal tap done, they have them curve into a fetal position to do it. And they stick a needle between L3 and L4 and pull out a small amount of the fluid. What they make them do afterwards is they make them stay in this fetal position for the next at least a few hours. I don't know how long they do it, but they want them to stay like that because they can have severe migraines afterwards if they get up and start moving around. Because this is a fluid that essentially is cushioning, and the brain and the spinal cord are floating in it. And it takes time for it to be produced. So once you take away a little bit of it, you have to replace that. Your blood needs to be able to replace that volume of it before it's not going to negatively impact your body. And it's a pressure thing. So you take a little bit out, and then your brain and your spinal cord don't have as much fluid to float on. And so your body needs to recreate that cerebrospinal fluid before you're going to be able to walk around without some negative repercussions. Questions?
I just got a question. So like when you have a C-section and they do a spinal block, is it in the same area?
I don't know.
Okay. I was just curious.
I want to say it probably is. Can I look into it a little bit before next time?
I was just curious.
Well, Rachel, I'll still look into it because I'm curious now too. I don't
Okay.
know. If my mom was here, she could probably tell you, but I she's not here, so and I don't know. So give me a little while to look into it.
Okay.
An epidural injection is when the needle is inserted into the epidural space, but it's going to be, remember, the epidural space is above the dura mater or superficial to the dura mater. So it remains, the needle is inserted into the epidural space, but not up into the dura mater. So anesthetics are sometimes introduced into this space to block pain signals during childbirth or surgery. And yes, I'll look into what that spinal block is because I know I've heard of those, but I don't know what they do. Do you remember we talked about white and gray matter before?
Yes.
So the central part, this is looking at a cross section through the spinal cord. The central part of the spinal cord is gray matter. And it's kind of shaped like a butterfly. Here's the anterior surface. Here's the posterior surface. So this is where your spinous processes of your vertebrae would be. Be right back here. You have an anterior horn, sometimes a lateral horn, and a posterior horn. The gray matter is here, the white matter is out here. So the gray matter, just like we talked about all gray matter is neuronal cell bodies with very little myelin. This is the site of information processing and synaptic integration. White matter is abundantly myelinated axons. They carry signals from one part of the central nervous system to another. Bundles of tracts, remember tracts in the central nervous system are bundles of axons. Nerve fibers or nerves are bundles of axons in the peripheral nervous system. So because we're referring to the spinal cord, bundles of axons, tracts, carry signals from one level of the central nervous system to another in the white matter of the spinal cord. The gray matter in the spinal cord has a central core, gray matter, that looks like a butterfly in cross section. There's a pair of posterior horns that carry the posterior root of a spinal nerve, which contains sensory fibers or sensory neurons, and the cell bodies of interneurons. Or remember, another word for the cell body is a soma. There's a pair of thicker anterior horns that carry the anterior root of motor fibers. Or we can also call it the ventral root, dorsal root, ventral root. The gray commissure, this area right here, connects the right and the left sides of the spinal cord. It has a central canal that's lined with ependymal cells. Remember what ependymal cells are. So these are neuroglia in the cerebral, in the spine, in the central nervous system that secretes cerebrospinal fluid. And a lateral horn, which is visible from T2 to L1. The lateral horn contains the cell bodies of motor neurons of the sympathetic nervous system or the visceral or autonomic nervous system. So, and posterior horn is going to carry sensory nerve fibers with the cell bodies of interneurons. Anterior horns are going to carry the motor fibers of motor neurons. The lateral horn, which is only seen from the T2 to the L1, is going to carry the cell bodies of motor neurons of the sympathetic nervous system. Stay with me for just a few more minutes here. The white matter of the spinal cord surrounds the gray matter. So white matter surrounds gray matter. Remember this, white matter, gray matter inside. It is the bundles of axons that go up and down the spinal cord, allowing for communication between different levels of the central nervous system. These bundles are arranged into 3 pairs of funiculi, or that are columns. There's the posterior, lateral, and anterior funiculi on each side. There's the anterior funiculi, lateral funiculi, posterior funiculi. The subdivisions of each of the funiculi are tracts or fasciculi. All the fibers of a given tract, remember of a funiculus, fasciculi, all the subdivisions of a given funiculus, tracts, or fasciculi. All of the fibers of a given tract have a similar origin and destination and function. Ascending tracts are going to carry sensory nerve impulses up the spinal cord toward the brain. Descending tracts are going to conduct the motor nerve impulses down the spinal cord from the brain. Remember where the sensory neurons are coming in through the posterior horn. And the ascending tracts are taking the impulses from the spinal cord to the brain. Descending tracts with the motor nerves are going to come out, are going to exit the spinal cord via the anterior horn. And the descending tracts in the white matter are conducting the nerve impulses down the spinal cord from the brain. So the sensory motor tracts in the spinal cord are continuous with sensory motor tracts in the brain. You guys, do you have questions for me? We'll stop here and talk about spinal tracts. And we'll cover some of this again on Thursday. Have a wonderful 2 days.
Thank you, you too.
Thank you.
Thank you.
Welcome.
You.
So I was able to see my answers for the exam, but
Okay.
I'm still not able to see the LabQuest answers. I think that's what I meant
Well,
to say earlier.
which one? I can't show you all of them because I have a lot of people who have not done LabQuest 2 and 3.
Oh, okay.
I know,
So
and that makes it really hard. It's the kind of the adventures of an online course. I give me 1 second here, okay? I wish, I can't, it's hard for me to show you answers for those exams because some people are yet to take them and I don't, and with an online course, as a professor, I've learned that you have to be really flexible about a lot of deadlines.
gotcha.
But I can't start showing you the answers for those without most of the people having taken them. Does that make sense?
It does. But I was just curious, like, why do I keep getting crap wrong?
Yeah, I don't know what to tell you. Give me 1 second though. Let me look, okay? Okay, I can get you I can show you the, bear with me just for a second here. Yeah, I mean, for example, let me just tell you. So for the second LabQuest, of the like 24 people we have in this class, 1/4 of them have not taken the second LabQuest.
Oh,
and I
okay
can
then.
almost assure you that those students are going to recognize that their grades are down for that, for the overall course, and are going to be asking me to open that up for them again. But I can, everyone has taken the first one.
Okay.
And I can show that to you. Give me 1 second here. I'm going to share my screen with you again. Yeah, I've learned a lot in online courses. You have to be really, a lot of times you have to be lenient with deadlines. Because a lot of students are, I can understand the internet connections make it hard for students. And there's a lot of different adventures that make online classes different from face-to-face classes.
Yeah, this is my first time doing online and I'm definitely not a fan.
I'm sorry.
No, I just like being in person.
You know what? And I understand that too. I have a preference for it too. I don't mind not having to be in my car all the time. I teach face-to-face at the other 2 schools that I teach at. And
See, that's what I thought I was signing up for when I signed up for this class, honestly.
I would, if you like face-to-face, make sure you sign up for a biology 122 face-to-face. And I think go ahead.
I was about to say, because that's what I thought I signed up for with you, because it said like York campus and it didn't say anything about virtual or Zoom or anything when I signed up for the class.
Whether or not it's remote?
See, I don't even know. I just said York campus. So I was like, oh, I don't have to go very far. So that's what I clicked on. But it's okay.
All right. Rachel, before I thought I saw your name. I'm going to stop sharing my screen again. Sorry. Stop sharing my screen again. Okay, 1 second. Let me look. All right. I can't see where I saw it the other night. Oh, here. Give me one moment. Okay. I was just going to make sure that you weren't signed up for my 122 class next semester. Because if you don't like online classes,
Yeah,
you learn that pretty quickly.
I didn't even know we could sign up for classes already.
You can, but do me a favor, if you don't like online classes, don't sign up for my next semester.
Oh, so you're online again?
I am. And I personally, because I teach, I don't know if I've told you this or not, I teach at 3 different schools. My other 2 schools that I drive for.
Mm-hmm.
And for me, this is the only way that I continue teaching at Hack is to teach remote for them.
Gotcha.
Because it would be another additional 2 or 3 hours a week in my car. And I can't do that right now.
No, I understand. It's just that I thought I was signing up for at least one online. I mean, because my other 2 classes are online.
Yeah. There are lots of face-to-face classes.
I guess I got to pay attention when I'm registering.
Okay, what was I looking for? Oh, I was looking for feedback for LabQuest 1. Give me 1 second here. Yeah, I think I don't think you're unique in that you struggle with an online course, because I think a lot of people struggle with online courses. That's the reason that I try to be really flexible about deadlines.
And the sad part is I already taken this class, but it's been so many years because I have a biology degree.
Oh, wow.
But I graduated in 2010. So when I went back to school to do whatever I'm doing,
yeah,
my shelf life on all these classes were expired. So I have to do it over again, which is more like a refresh. But that's why I'm like, why am I like, I cannot get an A in any of the LabQuest or exams or quizzes. So it's frustrating.
Yeah. Well, and it's changed. Where did you get your degree from?
University of North Carolina at Pembroke.
Wow. And it's changed. What I took anatomy and physiology wise and what I teach now is very different.
Yeah, because some of the stuff that we're going over right now, I don't really remember. Like I must have spaced out or something.
It's constantly expanding. It's constantly expanding. And, in our understanding, my mom took anatomy and physiology. She's a nurse practitioner.
Mm-hmm.
And she, I'm going through stuff that she's not heard of.
Right.
But one thing that she can do that I don't understand is I've never worked in patient care. And so she can tell me about things that are so bizarre. And then I go, we discuss it because she talks about it from a healthcare perspective. I talk about it from a physiology perspective. Like, okay, you saw this with the patient. Well, how did that occur?
Right.
And so I think it's always expanding. What I learned in physiology and what I'm teaching you are very different.
Yes.
And I'm guessing I probably took, if you graduated in 2010, that's probably about the same time that I took my first anatomy and physiology course.
Yep, I did. I did all that hard work and now I'm doing it all over again.
But see, look at how amazing your head is going to be by the time you're done here.
If I have one.
If it's not complete mush.
Yes. But thank you.
All right. So about 2:30, about 12:30, you should be able to open it up. It should be, okay. You should be able to look at them.
Okay. Thank you.
You're welcome. I'll talk to you soon. All right.
All right. Bye.
Bye-bye. Courtney and Michaela, are you there?
Yeah, I'm just trying to see if you received my lab quest.
When did you send it?
I'm pretty sure I did them, but I'm not sure because I know sometimes I know sometimes you won't receive
well, sometimes, yeah, they just don't go over.
They don't go through.
This is Michaela, right?
Yes.
Okay, give me 1 second to look. So I'll be able to tell you. Michaela, what's your last name?
Costly.
Say Costly, I remember now. Yeah. Okay, I don't have a score for Lab Quest 2. Okay. Did you do Lab Quest 3 too?
Mm-hmm.
I don't have a score for that one either. Do me a favor, can you send me a screenshot showing you completed it and what the score was?
Mm-hmm.
And then I'll update the grades.
Okay. All right. I can send them. I'll send them now.
Okay. Perfect. It may be at least an hour before I update your grades because I have class in about 5 minutes.
No, that's fine.
Okay. Mm-hmm. But if you don't see them updated by the end of today, like by 10 o'clock tonight, because I do have a class later today at a school. Mm-hmm. If you don't see them updated by 10 o'clock tonight, email me and remind me again.
Okay.
Okay.
All right.
I'll talk to you soon, all right?
Yes, you too. Have a blessed day.
You too. Bye-bye. Courtney, are you there?